I'd like to discuss with you today is this work that I did during my postdoc, and I continued after I joined Mount Holyoke College. I'm interested in memory, and in particular, I have been studying the role of the entorhinal cortex in hippocampal physiology, but also in how the entorhinal cortex plays a role in memory. So I will give a brief background and introduction so that you can understand the different techniques that I used. I will talk about the research question and how I was experimental design, and then I will tell you about my results, some conclusions, and future directions of what we are doing right now. So I'll start by talking about why memory is interesting to me, and I think that you will agree that memory is a very fascinating concept, because we are who we are because of the things that we can remember. And when we remember things, we remember not only what happened, but also when those things happened and where they happened. So we have a spatial component in our episodic memories, and we also have a temporal component. But how does the brain do this? In 1957, Scoville and Milner reported a very extraordinary case, the case of patient HM. I'm sure that many of you heard about this case. So HM had a bike accident when he was 9 years old and sustained a brain injury. And as a result of this brain injury, he started to have a lot of seizures. His epilepsy got really bad. And at the age of 21, he was having a lot of seizures. So they decided to take out the part of the brain that was causing these seizures, that was the medial temporal lobe. And in the medial temporal lobe, the main structures are the hippocampus and the entorhinal cortex. So when they did this surgery, they helped a lot patient HM with his seizures. He was not having epilepsy anymore, but they left him with a very profound amnesia. So he could not form any new long-term memories. And so Brennan, Milner, and others proposed that the medial temporal lobe was required for memory. And evidence supporting this claim comes from results of different tasks that patients do, memory tasks. So what you are seeing here is the performance of 2 patients that had hippocampal lesions in this complex figure test. So what the patients are asked to do is to first copy a very complicated drawing. And you can see that when they are copying the drawing, they do a very good job. It's very similar to what a control patient would do. So they have no problems in perception. But if you take the drawing away and wait a few minutes and you ask them to recall that, they have a lot of trouble recalling what they saw before. So they have a very big problem, a memory problem. You can see here the performance of a control patient, and you can see that the hippocampal lesion patients have some problems in recall. But even though definitely there is an impairment, you can also see that there is a sign of some remembering. So they remember a little bit. However, when the lesions are also including the entorhinal cortex, they are complete medial temporal lobe lesions, there is no recall whatsoever. And if they are like, if you insist for them to draw something, they would draw something that is completely unrelated to the drawing that they were exposed to. So which are the neural computations that support this memory? In the 70s, there was a very big step towards our understanding of how the brain is able to form new memories. So John O'Keefe discovered these cells that were called play cells. And what you are seeing here is an animation in which an animal, a rat, is running around an open field. The path of the animal is in gray, and in red is every time this particular neuron that O'Keefe was recording fired an action potential. So you will see in red all these action potentials, and after a while you will notice that there is a particular part of the field that is coded by this neuron. So what he found and what he proposed is that there are some cells in the hippocampus that are able to map specific parts of the environment. And these are called place cells. So we will have a place cell that is coding this particular part of the environment. Then there will be another cell that fires when the animal is in a different part, another cell that fires when the animal is in another part. And so together, all these cells would form a map. And this is very important for navigation, but it's also very important for memory, because as I said, there is a spatial component of memories that we use to put into context what happened to us. And together with O'Keefe's findings, the Mosers, Maybrit and Edvard Moser found what they called grid cells. This time they recorded in the entorhinal cortex. The entorhinal cortex is the main input to the hippocampus. So information from the entorhinal cortex is probably very important for hippocampal place cells. So they wanted to know what is happening in this other structure that is projecting to the hippocampus. And they found these cells that they called grid cells. They call them grid cells because you will see that the firing pattern is very similar to a grid. So they, instead of having one place field, they have several fields where they fire action potentials. And it seems like they are very important for estimating distances because these different patches are equally distant from each other. So together, John O'Keefe and the Mosers won the Nobel Prize in Physiology or Medicine in 2014 for their discoveries related to self-location, but also to memory. So we have a pretty good understanding of how space is coded in the brain. As I said, the medial entorhinal cortex, I'm gonna also talk about it as MEC, projects to different parts of the hippocampus. And so these grid cells seem to be helping the hippocampus to form place cells. But memories don't only have a spatial component, they also have a temporal component. And so less is known about how the temporal context of episodic memories is formed. In 2008, another group reported what they called sequence cells that later were called time cells. And these cells fire in a sequence when the animal is waiting. So imagine, for example, this animal is doing a memory task, and between trials, they have to wait for, let's say, 15 seconds. And during those 15 seconds, there are different neurons in the hippocampus and in other brain areas that fire in a sequence. And the interesting thing is that this sequence is always the same. So when the animal comes back in the next trial, the same cells are going to fire in the same sequence. So what you are seeing here in the different panels are examples of different cells. So this is one cell that fires at the beginning of the intertrial interval in trial 1, and in the same place in trial, or the same time in trial 2, the same time in trial 3, and so on. This other cell fires more towards the middle of the intertrial interval, and that's what you are seeing here in form of a heat map. So the warm colors are going to indicate that the firing rate or the action potentials are more. And so you can see that all the action potentials for this particular neuron always happen at the same point in time. And for this neuron, they always happen towards the end. So at the end, we will have, again, a sequence of cells that fire in a similar manner, trial through trial. However, it's less clear whether the medial entorhinal cortex is important for this temporal firing in the hippocampus. And it's also very controversial whether or not these time cells are required for memory, because we do know that there is this sequence that is observed during intertrial intervals, but it's not clear whether that sequence is needed for memory performance. So that's what guided my research and what this project is about. I was interested in knowing whether the medial entorhinal cortex was required for memory performance, and also what were the physiological correlates of this memory performance. So if the medial entorhinal cortex is lesioned, do we still see time cells in the hippocampus? And if not, are these time cells, impairing memory performance? So by investigating the physiological dysfunction on these animals, our hope is to provide insight into network mechanisms that could be either enhanced or regenerated in disorders that have, that involve damage in these circuits. Okay, but how can we do this in rats? We cannot ask rats to draw a very complex drawing, unfortunately. So we had to think about a task in which we could see this memory performance. And we thought about the alternation task. So in this task, what animals have to do is to keep track of different parts that they visit. The different contexts are very similar. The two arms of the maze are very similar, but they have to be able to differentiate where they are so that they know where is that they want to arrive in order to get the reward. And so basically, this is kind of an idea of what we did, but really what we did was much more simple. We had animals alternating in a figure-8 maze. So they had to go, let's say, in the first trial to the left, and then wait for a delay that was variable. I will tell you that in a moment. And then after that, they will have to go to the opposite side. So they had to keep this information in mind of where they went in the previous trial in order to succeed in the next one and go to the opposite side. Okay, so they just had to alternate. This is very easy for animals, and there is not a lot of working memory load, because when you do this in a pattern and the animals run these tasks very fast, if there is not inter-trial interval, they don't rely on the hippocampus. They can do this as a habit and using other brain systems. However, if you stop the rat in the middle here in the delay side for either 10 seconds or 60 seconds, then the task becomes hippocampal dependent. So the animal needs the hippocampus in order to keep in mind the information of where they went in the previous trial, so that they can remember that and go to the opposite side in the next trial. So we were interested in knowing whether this task was also dependent on the medial entorhinal cortex. So what we did was to lesion the entorhinal cortex and then train the rats first without any delay between trials. So they just had to learn the pattern, the 8 pattern. And then we tested them also inserting either 10 seconds or 60 seconds between trials. We implanted electrodes to see what was happening in the hippocampus while the animals were running this memory task in vivo. And also to see whether the medial entorhinal cortex provided information to the hippocampus that was relevant for this task. So we implanted the electrodes, and we had the animals doing the task again, and then we did histology to see where our electrodes ended and also to see how good our lesions were. Okay, and so this is a picture of what the medial entorhinal cortex looks like in a control animal. So these layers here are the medial entorhinal cortex, and this is the hippocampus in 3 different sections. So our lesions were aiming those areas. And what you can see is that we were pretty successful targeting those areas and not the hippocampus. So animals lack the inputs from the medial entorhinal cortex. And what we saw is that when there is no delay between trials, both control animals and animals with lesions in the medial entorhinal cortex perform the task very well. So this is the percentage of correct choices. You can see that they are above 80%. across all days, all phases of the experiment. However, they are a little impaired at the 10 seconds delay, but especially impaired at the 60-second delay. So when you have the animal wait for 1 minute between trials and they have to keep the information about where they came from during that minute, they forget and they are not able to perform as well as controls under this condition. You can see that towards the end, we also find that there is a difference between control and emission animals in the 10-second delay condition, but we believe that this is mainly driven by the improvement of the control animals that get really good at it, and you can see that the performance is better. But the lesion animals do not improve much. However, there is a little bit of an improvement in the 60-second delay. As you can see, they start at chance levels and they improve. So we were not only interested in seeing what were the physiological correlates of the memory impairment, but also how could they improve? What was happening at the physiological level in the hippocampus that maybe helped them to improve in this task. So before I show you the electrophysiological results, I just wanted to give you a very fast crash course in vivo electrophysiology, because this is a complicated technique and I'm gonna show you some graphs that might be confusing. So I first wanna very quickly explain how this works. So what we do is to record action potentials of individual neurons. And to do so, we build these electrode arrays. Each one of these channels or legs have one electrode, and all those electrodes are individually moved down to our target locations. The only part that touches the animal's brain is this tiny part at the end. So the animal wears this as a hat, but it's a permanent hat. And each one of these electrodes, as I said, can be lowered individually. Each electrode is formed of 4 channels. This is like thinner than a hair. And each electrode has 4 channels, okay? So that's why we call them tetrodes. They are called tetrodes because they have 4. And it's very important that they have 4 channels because it allows us to compare the information or the voltage deflections that arrive to each one of the channels so that we can individualize the different neurons. And this might sound a little complicated, but it's very easy. So just bear with me, and I'm gonna try to explain how this works. So imagine that we have one tetrode, okay, in the hippocampal cell layer, and we are recording the red neuron. Okay, so the red neuron is going to be firing action potentials. Every time the neuron fires an action potential, those differences in voltage are going to arrive to our electrode. But the voltage deflection that is going to arrive to, let's say, channel 1, okay, this is channel 1, this one here, is going to be a bigger deflection, right? The amplitude is going to be bigger than what is going to arrive to channel 4, because channel 4 is farther from the red neuron, right? And so we are able to then compare the amplitude in channel 1 with the amplitude in channel 4 and know that all these action potentials are coming from one specific neuron that is closer to 1 than is to 4, and the opposite will happen with the blue neuron. We compare what the action potentials that arrive to each one of the channels, and we are able to plot that information comparing, for example, the amplitude of the voltage deflection in 4 versus the amplitude in 1. And all the action potentials are going to cluster together. And we know then that this is coming from one individual neuron, while this is information from a different neuron. So we do this many times with all the neurons that we record, and we are able then to know what is that each specific neuron is coding for when the animal is running the task. So in this animation, what you are seeing is the action potential of one particular neuron while the animal is doing this memory task. You can see that this particular cell fires more when the animal is passing through this side of the maze versus this other side of the maze. So in order to facilitate the view, we plot the information in rate maps. You are going to see a lot of this in the results. So rate maps are basically heat maps in which we color code the action potential. So when there is a lot of activity, we put it in a warmer color. So this means that this particular neuron fires the most here. And we can also put this information in form of a linear graph. So here you're seeing the same information that you're seeing here, but now we are just putting it in a line. So we are indicating that this is the return arm. This part here represents the delay. This part here represents the stem, and this represents the reward. Okay. So now I'm going to show you the data. What we found was that when you lesion the medial entorhinal cortex, you find bigger place fields in the hippocampus, and they are more unstable, meaning that when the animal is running around, these place cells move and they are not accurate in telling the animal where they are. So we wanted to know more about this and exactly like in which parts of the task there was an impairment. So what we did was to separate the different parts, because the animal is doing different things. For example, when it's coming back from this side, we call that encoding, even though this is a working memory task. So I know that is not really encoding, but we call it encoding because this is the phase in which the animal needs to keep in mind, is the phase in which the animal needs to differentiate whether it's coming from the left or it's coming from the right. So it's the encoding phase. And what we can see is that animals that don't have any lesion have beautiful place cells. What you can see here is that trial to trial, in this side, the cell doesn't fire. So you see that there are no action potentials. But when the animal is coming from this other side, the cell, this particular cell that's an example, always fires in the same place. And presumably this is giving the animal information about where they are so that they can perform the task. What we found is that if you lesion the medial entorhinal cortex, these cells are not coding information in a reliable way anymore. So what you see is kind of a mirror image of what the cell is doing the same thing when the animal is coming from the left than when the animal is coming from the right. So this cell is not providing any useful information to the animal because it does the same thing when the animal comes from one side and when the animal comes from the other. And so we think that maybe this is in part explaining why they are impaired in the task. What was very interesting and a little bit shocking to see is that animals that have lesions in the medial entorhinal cortex have a preserved firing in this other area of the hippocampus. So the hippocampus has different areas. The CA1 is a big part of it, but then there is another part that is called CA3. When we record neurons in CA3, we see that they actually fire similarly to how control animal cells fire, so coding only for one side of the maze. So we were very confused about this, and how we interpret this is that, or how we want to think about it is that maybe it is because the entorhinal cortex projects to different parts of the hippocampus, so that the CA3 area is receiving inputs from different parts. And it's also a very recurrent network that projects to itself. So maybe that's why the CA3 area is less impaired than the CA1 area. But we wanted to see whether that impairment was still there during the retation phase. So in the retation phase is the phase in which the animal has to keep this information in mind, as I said, during the delay. And before I told you about this sequence or time cells that fire in the same way, trial to trial. So cell A will fire first, then B, then C, then D. And then in the next trial, the same thing will happen, okay? But what I thought is, Well, the animal is coming from different sides. So this sequence would make sense to be different, right? If the sequence is the same, then how is this sequence telling the animal anything important about the task? It should be a little different, right? When the animal is coming from the left or from the right. But it is also true that the left and the right side of the maze are very, very similar. So the difference should be very subtle and it would be hard to find. So I like to think about this, and I like to give this example of how time cells work and how this sequence seems to be always repeating and always being the same. And I like to give the example of our daily routine during a pandemic, especially when we were in lockdown. I don't know for you, but for me, every day seemed to be the same. So for example, I would wake up in the morning and go pick up my Amazon package. Then I would play some puzzles. This would be me trying to figure out what to watch next on Netflix because I already watched everything. This is me also realizing that I still have another 8 Zoom meetings to attend, and it's only 10 a.m. Buying some essential items for the week. So this sequence would repeat and repeat every day, and every day seemed to be the same day. It's kind of a similar idea how these time cells work. But what I was thinking is that if I know that today is Thursday and it's not Tuesday, something had to be different, right? And so something in the sequence should be different too. For example, maybe instead of going grocery shopping today, I go for a bike ride. That was what I was trying to look for and to find is in these time cell sequences. However, to my surprise, what we found is that the sequence for left trials and the sequence for right trials is very, very similar. It's so similar that even if you try to train an artificial network to guess whether it's a left or a right trial, not even the machine is able to tell. Okay, so if an artificial machine is not able to tell, it's going to also be very hard for us or for the mice to figure out when it's a left trial and when it's a right trial if we only pay attention to this sequence. And this is in controls and also in lesion animals. So not even control animals differentiate between left and right, or I should say not time cells differentiate between left and right. And another thing that was interesting is that even though there is a little bit of a sequence, this sequence that doesn't last for more than like 15 seconds. After 15 seconds, all sequences disappear in both control and lesion animals. This was a little shocking because if you are familiar with the literature, people are really crazy talking about time cells and how important they are and how interesting they are. And we were finding something totally different. But no one before had recorded for more than 15 seconds, and we recorded for 20-second delay intervals. So this was very interesting, but also a little scary to report because we were going to basically say that these time cells were useless when everyone was loving them. But then we were like, okay, if there are no time cells during the retention period.
5 minutes, Professor.
5 minutes, okay, thank you. If there are no time cells there, how are the animals able to do the task? So what we saw is that even though there is no retention during the delay, or there is, I shouldn't say there is no retention, there is retention for sure, but it's not in time, is not something that the time cells are carrying. So what we see is that this information comes back again when the animal is about to take a decision and turn in the right direction. So you can see that this cell only fires when the animal is going to go left, but not when the animal is going to go right. And I'm just pointing to this trial because this is a very interesting trial in which the animal was going to go to the left, and so the cell started firing, but then the rat turned and went to the right, and the second they turned, the cell stopped firing. And I just find this so cool. So I wanted to show you. So what we think is that maybe CA1 is helping CA3 is helping CA1 to remember, because what I have said, and I know that there is a lot in here, but what I have said is that initially CA1 is not able to tell, or cells in CA1 are not differentiated between left and right trials, but after the delay, both the CA1 and the CA3 network differentiate between left and right. So what we think is that maybe CA3 is sending some information to CA1 so that they can both help animals at this point of the task. And the take-home message is also that this information is not retained in time cells during the delay interval. So what we think is that maybe it's retained in another way, for example, in form of a short-term synaptic plasticity, but we don't know this yet. So the open question or the future direction is like, do these time cells represent the lapse time in the hippocampus? Because this task is not really assessing the time is not an important variable. So what we are looking at now is what happens when the animals have to actually take a decision that is based on time. Does this sequence differentiate them between 10 seconds and 20 seconds? And does it go farther? Like, is it longer than 15 seconds? So that's what we are doing right now. And we have some preliminary data that shows that MSC lesion animals are not able to differentiate 20 seconds delays, but that's a different story. And I want to finish. So I want to say thank you to everyone that participated in this research. A lot of this was done in the Loikab lab. That was a place I was doing my postdoctoral research. And these are some of my students that also helped to collect the data. So thank you very much. And thank you for your attention. Let me know if you have any questions.